Okay, good morning. How are you? Morning. Before we start, do you have any questions to me about what we talked about last time or anything that everyone else could not answer about MS stats or linear models? Well, I'm sure we'll have questions as we go, so please don't hesitate to interrupt me and even ask questions about what we did previous days. Really doesn't matter. So what I would like to do today in the hour and a half that we have is to talk about a collection of methods, many of which are inspired from genomic literature. So what we noticed when we are working with our collaborators or when we review manuscripts is oftentimes people take methods which are developed for high-throughput, large-scale data other than mass spectrometry and proteomics, and in particular, microarrays and RNA-seq are clearly a motivation. And then these methods are applied as is to quantitative proteomic experiments. And so what I would like to do is to comment on how that fits and what is appropriate and what is maybe not as appropriate, and how these existing methods can be leveraged in our context. So I have essentially a collection of different things which are used in various contexts. And so we will see how far we get to that. And this will include extensions such as the analysis of TMT experiments. So this is the plan. So first, a very common approach which people use. Okay, so the first very common method that people refer to, especially when we talk about problems that we discuss here, such as class comparison, right? So comparison of average protein abundances across conditions, is to use a method called LIMMA. So LIMMA stands for linear models for microarrays. And this was one of the first methods for testing for differential abundance, which was developed for microarrays, and it was quite successful and quite influential in the statistical literature. And so I see that oftentimes people are using these methods also for quantitative proteomics. So just to remind you, the goal for us is to compare the abundances of protein in the underlying population. So we have two sources of variability, and our goal is to take the values which we observe, in our case, the intensities of mass spectrometric peaks, and use these values to make conclusions about what we do not observe. So these are the differences in the average abundances in the underlying populations. And as before, we work on the log scale, right? So we talk about differences, thinking of them as estimates of log fold changes. And one issue that is important for us is the bias. So when the differences that we observe are systematically different from the true changes. And the other aspect, and this is what we would like to focus on, and this is what LIMMA is focusing on, is the uncertainty associated with our summaries. So essentially, when the experiments have too much noise and uncertainty, then the variability of our summaries is too large, and we cannot really say anything meaningful about the differences in abundances in the underlying populations. And there are many reasons why this uncertainty can be large. It can be because our, the natural abundance of proteins is quite variable between individuals, and then it's hard to make conclusions about the underlying populations. But another very common reason for large uncertainty in the summaries is the small sample size. Oftentimes, we cannot afford a very large number of biological replicates, especially if these are pilot studies or initial screening experiments. And if the experiment has a small number of replicates, then we know that each mean in each group can have quite a bit of variability. So if we repeat the experiment with the same number of replicates, we may have a somewhat different value. And therefore, we have a lot of uncertainty in the signal, and this prevents us from making conclusions about the underlying populations. And LIMMA specifically addresses that. So how can we make a better inference, better conclusions in experiments which have a limited number of biological replicates? To be clear, LIMMA does not address any other things. It really specifically addresses the uncertainty in our estimates. Let me also remind you of what we talked about in the beginning in a very simple case, and let's just stay with this very simple case. So let's say we have a label-free LCMS experiment where we have one peak per protein per person, or maybe we have more peaks, but we have some ways of summarizing this so that we have one number per protein per person, and we compare the abundances of subjects in this group versus this group, right? And so what we did for that, we talked about this quite a bit last week, to distinguish systematic changes in abundance from artifacts of random variation, we essentially constructed the signal-to-noise ratio, where the signal is the difference between the means in each group, and the noise is the variability associated with the individual means. If you were not with us last week and you don't know this, let me just say very quickly that this quantity in denominator is the variance of the mean of the first group, so it's called the variance of the sampling distribution in the first group. This quantity is the variance of the second mean, and then we take the square root of that to have it on the same scale as the measurements. So this whole denominator is called the standard error of our signal. And we talked about this quite a bit last week. If it is not clear, we have videos already on YouTube from last week, so just take a look and refresh your memory. What I didn't really say clearly last week, but now we need to add this, is that if we assume that there is no difference between the two groups, then we know the expected range of values that the signal-to-noise ratio can take, and this range of values follows a student distribution. The student distribution has a parameter called degree of freedom, so this distribution is characterized by this particular parameter. So this is the number of data points in the first group plus number of data points in the second group minus 2. 2 stands for the fact that we calculated two means to create the signal. So we used 2 data points to, so essentially we calculated 2 numbers from this data. One number is the mean in the first group, and the second number is the mean in the second group. And if this parameter increases, so if the sample size in each group increases, this distribution becomes more and more similar to the normal distribution. Okay, so that's really what we talked about last time. I'm just adding here this quantity called degree of freedom. Again, to refresh your memory, if the null hypothesis is true, so if we assume that in fact there is no difference between the two groups, if we assume that each individual data point is independent and follows normal distribution, then we can mathematically claim that the range of the values, or the values which of the signal-to-noise ratio, will follow this student distribution. And again, if we have a small sample size, it means that N1 is relatively small, N2 is relatively small, and therefore the denominator is relatively large. This was one issue, and the other issue was the fact that the small sample size gives us difficulties in estimating the variability in each individual population. We looked at this figure last week, which comes from the points of significance collection in Nature Methods, and it illustrates the challenges of the sample size. So they started from a distribution which is very non-normal, so it looks some kind of weird distribution, and they take data points from this distribution, they simulate this of different sample size. So here the x-axis is a different sample size, and they calculate means, standard deviations, and standard error of the mean, right? So just to remind you, standard deviation is just characterizes the variability in the population. Standard error of the mean is the square root of this quantity, so how confident we are in the mean. And what we said was that as sample size increases, the sample mean will vary, but as sample size is large, it will be very close to the true value. Sample standard deviation will do the same, but, and this is really the important point for us, when the sample size is small, you see that the estimate of standard deviation doesn't fluctuate on both sides of the true value. It actually tends to be below the true value. And the reason for this is that when we have a small number of replicates, not only our quantities are unstable and not reproducible from one experiment to another, but they also we don't have enough data to have a full sense of how much variability we have in the underlying population. So for example, here, if we take 3 data points, it's possible that all the 3 data points will come from this range of values of this probability distribution. And we will think that this is how much variation we have in our population. But in reality, we have a lot more variation because we just didn't have a chance to sample any data from this end of the distribution. When sample size increases, will sample all the plausible values with high probability, but when sample size is small, we may end up just in one corner of this distribution and not be able to see the full range of possible values. So the problem with sample size is not only that our means are unstable and not entirely reproducible from one experiment to another, but also we are too optimistic in terms of how much variability we have. And therefore, we can either not find changes when they exist, or we can be too optimistic and think that the variability is too small and actually claim changes when the changes are not present. So we can really make mistakes in both ways. We can have many false negatives, but we can also end up with many false positives. And this is really the problem that LIMMA tried to address. So it came out in the very early days of microarrays, where the technology was fairly expensive. Usually people had small number of replicates, and this method attempted to address this issue. Another thing that we discussed also last week is that one way to essentially have more data in situations when replicates are rare is to add more conditions. So we can add more conditions from the same problem. Sometimes it is difficult to have all subjects with the disease, but maybe we can have more types of controls, or maybe we can have more related types of stresses, which are maybe not the same stress we're interested in, but they may be cheaper stresses or easier to obtain. In some cases, it is easier to have replicates from related conditions than more replicates from exactly the same condition. And in this case, what we can do is, so here it's kind of disappeared, but if we have more than two conditions, we can have subjects from different groups. Each group will have its own mean, of course, right? Because it handles, it assumes that we have different extent of protein regulation in each group. However, the variability around the mean in each group may have something in common. For example, we can say that the variance is actually exactly the same in all the groups. Or we can say something like the variance is a function of the mean, and this function is the same across all the groups. Essentially, we can make some assumptions which would allow us to learn more about the variation in each group by combining information from different groups. This particular, so LIMMA, this particular approach assumes that the variance is exactly constant. Instead of looking at the sample variance of group 1 and sample variance of group 2, LIMMA assumes that the variance in each group is the same. Is it a good assumption or not? It depends, and usually it is a good assumption for some proteins, and it is not a good assumption for some other proteins. But this is the assumption that we use, and oftentimes it is necessary if the sample size is small, because we will not even have enough data to estimate the variance in each group separately. So this is what happens, right? So now we will modify the signal-to-noise ratio. This is a very common, and this is, by the way, not just LIMMA. You can do it without LIMMA as well. So now we will assume that, in fact, S1 and S2 are the same. So both groups have the same variance. And in this case, we will have essentially a consensus estimate of variability in each group. So we will give more weight to the group which has more replicates, and we will have a single constant which represents the variability into groups. And now, if S1 and S2 are the same, it kind of simplifies like that. I can have the S in front, and then we have the two sample sizes in the parentheses. So this is the modification to the signal-to-noise ratio that we discussed earlier, but in the case when we assume that the variance in the two groups is the same. So this is a little bit nicer already, because instead of having two variances in each group to estimate, we only have one variance. So we already pooled some information from two groups to improve the variance estimation between conditions. We hope that we will a little bit alleviate this type of situation, but this is not quite enough. And now what LIMMA is doing is the next step by saying, well, even if we pool the information about the variance from multiple groups, we may still end up with the number of replicates too small and the estimate of variability too small. What else can we do? And now, and this was really the main insight from the from this approach is to recognize that, unlike in many other statistical problems, when we have one data set, we have one signal-to-noise ratio, we have to make conclusions. Here we actually have many proteins, or in that case, many genes in microarrays. And in fact, what we are doing, we're fitting the same model and estimating the same type of variance parameters over and over and over again for every protein. Just like in MS Stats yesterday in the hands-on part, you essentially fit the same model over and over for each protein, right? And there was a p-value 1 by 1 for each protein. Same thing here. What we can do now is to, in addition to p-values, we just have many variance components for each protein. And the assumption that LIMMA is making is that all these genes or all these proteins in our study, well, they come from the same biological organism. They are quantified using the same measurement technology. So even though the proteins have very different function and different regulation, they're more related to each other than if we say take some other species, right, or some other biological organism and use some other measurement technology. And the assumption that LIMMA is making is that even though those variances are not the same, we can think of them as coming from the same family of probability distributions. And in practice, what is happening is that for statistical reasons, it's better to work not with the estimates of variance, but with the inverse of that. So they're looking at 1 over s squared. So this is our pooled estimate of variance between the groups. What they're doing, they're looking at the inverse of that. And essentially, you can make a histogram. You plot a histogram of those 1 over s squared across all the genes. And this was very novel at that time, because unlike in any other data sets, at that time, we never had many of those tests over and over across proteins. And now we'll see, well, okay, this is the histogram, so maybe some proteins are in this situation, their variance is too small. Maybe some proteins are in this situation, their variance is too large. But on average, they should be close to some kind of true consensus value. And of course, variances, they have to be positive, right? So the distribution tends to be quite skewed. And for statistical reasons, it may be appropriate to assume that this type of distribution follows some kind of a chi-square probability distribution. So it's a distribution which is limited to positive values, skewed to the right, kind of what we would see in these experiments. And what they actually do, they fit essentially a scaled version of the chi-square distribution to this histogram of 1 over s squared. And this fit of the histogram has two parameters, which they can vary to obtain a good fit. So one parameter is the average or consensus variance over proteins. So you can think of this as essentially expected value of this distribution. And the other is the average degree of freedom. So this degree of freedom is essentially an average quantity or consensus degree of freedom which would have for each individual protein. And so now we can say that we assume that overall these inverse variances will follow the distribution. We derive parameters from the data, and then there's an additional step where we can integrate the consensus information across all the proteins with the protein-specific information, and it looks like that. So this is what we had before. If we use pooled estimate of variability, and we essentially replace this estimate with a new estimate, which is a weighted average of the protein-specific information and the average information in the entire data set. So D is the degree of freedom, so how much data points we have for individual protein, and D0 is this parameter which we obtain by fitting a curve to this histogram. And you can see that if we have a lot of data, if D is large, so we have a lot of replicates in the first group and or in the second group, then this weighted average will give a lot of weight to the protein-specific variance. However, if D is small, then more weight will be given to the average variance across all the proteins. So essentially what LIMMA is doing is taking a middle ground in the estimation of the variability. So on one hand, you can say, okay, every protein has the same variance. That's probably a stretch, right? That's really likely not true. On the other hand, we can say each protein has its own variance, which is nice, except that we may not have enough data to really characterize this fully. And LIMMA is taking a middle ground and says we will have an estimate of variance, which is a weighted combination of assuming that everybody has the same variance and assuming that each protein has its own variance. And the extent of this weight is derived from the data itself. If you want to learn more about this, or if you know about this, this type of approach is called empirical Bayesian approach, or empirical Bayes approach. This type of curve is viewed as a prior distribution for our variance parameters, and it is empirical because the prior distribution is determined from the data set itself, as opposed to having some prior data sets or some other sources. This is what it's doing, and it is actually really nice, especially when the sample size is small. This is one of the first publications which describe this approach and compare this. This was one particular data set. I don't even remember where it came from. These are the volcano plots, just like we discussed yesterday. And you see that this was a volcano plot for the for just using a t-test, and this is using the approach in LIMMA. By the way, I didn't say that another word for this is using moderated summary or moderated test statistic. Again, moderation comes from the fact that it's a weighted average between protein-specific and consensus value. And you see that this is quite nice. So if we use the regular t-test, we can see, for example, that some large fold changes, they have very low, not significant p-values. On the other hand, you have some small fold changes which can have very small p-values. Improving the estimate of variability cleans it up quite a bit, right? So now we have more frequently large fold changes associated with statistical significance, small fold changes may be less associated with statistical significance. In other words, it really minimizes some of the artifacts which are due to the noise in the data, which is produced by the small sample size. And this is something which was very popular in mass spectrometry. What I have seen quite a bit is that people say, oh, okay, LIMMA was designed for microarrays. We have data structures which are similar as microarrays. Therefore, let's go ahead and use LIMMA as is. And I see this even up to date across the board, where people use LIMMA not because it can help with variability, but just because it has similar data structures. And I would like to argue that it's a good thing if you try to address the problem that LIMMA addresses. But it's not necessarily a good thing if the problem in mass spectrometry is elsewhere. And what we have in mass spectrometry a lot is multiple features per protein, missing values, and outliers, which was not quite, is not to the same extent was present in microarrays or in RNA-seq. And this is just an illustration. So here, let's see if we can, I really apologize for the quality of that. This is our IPRG dataset, which we worked with yesterday. So here we compare condition 1 to condition 2 with 3 replicates per group. And what this panel is doing, we take all the peak intensities on the original scale, take the sum, then take the log, and looking at the t-test versus LIMMA versus the approach by MS Stats. And these are volcano plots. It's a spike in mixture, doesn't look as nice as in the previous case. We can look maybe here. This considers all the changes which we find between this condition. Between LIMMA and MS Stats, we find 10 differences. Remember, there are only 6 spiked proteins, right? So there are some false positives here. LIMMA finds even more changes. T-test doesn't have much sensitivity. So this is what happens with the log sum. But now if we look at the summarization, which MS Stats is doing, and I really wish I could use Keynote, but then I don't have a mouse, so we have a problem. Now, on the other hand, if we use the summarization in MS Stats, which remember uses censoring and robust summarization, right, to have one number per protein per sample. And again, here's a t-test, here is LIMMA, and here is the MS Stats downstream statistical analysis. You see that LIMMA and MS Stats agree very well. They find 6 changes. T-test only finds 1 change. The conclusion that I have for that is that even though estimates of variation are important, the input numbers are just as important or even more important. So if summarization of multiple features for the protein is not quite done right, the moderation of the variance can only help somewhat. A good way to do this is to summarize the features, account for censoring, account for outliers and interferences, and then consider the experimental design. And if after all of that the sample size remains small, then LIMMA definitely helps. If after that these outliers and interferences are eliminated and the sample size is relatively large, then really there is no reason to add an extra complexity and extra assumptions in our model to improve upon that. And here's an illustration. This was another experiment which has more replicates, and you see that now LIMMA doesn't really make much difference compared even as a t-test. And the reason is that the number of replicates is larger. In my personal experience, beyond 3 replicates, this really doesn't make much difference. So it's really very, very small sample size when this moderated estimation of variance helps. In larger sample size, it is really not helpful. And we have many good reasons to have larger sample size. In our personal practice, we use it a little bit, but not as much, not as a default. Do you have any comments on that or questions? Does it make sense? Yes. Always, always, always. This is something we discussed last week. Let me just give you a short summary. Because we have 2 sources of variation, right? Biological and technological variation. We can have biological replicates, we can have technical replicates. Biological replicates are the most important. If you have 10 runs, you will be much better off having each run for a separate person, right? A separate biological replicate, as opposed to taking one person, measuring this person really, really, really well, but it's still one person. And the reason for that is that we're trying to understand what are the changes induced in the biological system more systematically than the natural variation in the biological system. And therefore, we need to have data to quantify the extent of biological variation. Yeah? Yeah. And we talked about, last week we also talked about how to show this mathematically, but just intuitively, I think it's pretty clear, right? If we want to say that the changes in biology are systematic, we need to know how much random variation in biology we have as well. Okay, you use p-value. Okay, same thing. We talked about this quite a bit last week. Take a look at the video on YouTube, but let me give you a very short summary. P-value is only appropriate when you have one protein in your study. Because the way the p-value is defined is it looks at the conclusions for a particular test of hypothesis, and it characterizes the uncertainty associated with the decision of this particular hypothesis. If you have multiple proteins, individual p-values do not characterize the uncertainty in the collection of the tests, so in the collection of proteins. Now, in our experiments, I don't know about you, I've never seen mass spectrometry experiments with one protein, right? So the whole point why we're doing it is because we want to test many proteins. Therefore, we need to make conclusions for the whole collection of hypothesis tests. And therefore, we need to control error rates associated in the whole collection of tests as opposed to the individual tests. And false discovery rate is one type of criteria which we can use to control the multivariate error rate. And p-values adjusted for false discovery rate are such that the cutoff of the adjusted p-value tells you what is the estimated false discovery rate in the list of claimed differences after this cutoff. To summarize, for the type of experiments we talk about here, we always use adjusted p-value, and we do not use p-values without adjustment. Take a look at the video, maybe come talk to me during the break, because this is a really important issue. And what we also said, and let me also repeat, I apologize to those who were here before, what I hear a lot is that people would do experimental design, they will collect data, they will process data, maybe summarize features into some kind of summaries for proteins, maybe have p-values, adjust p-values for multiple testing, and then they don't find what they would look for, what would expect to find, and then they blame adjustment for multiple testing. When in fact they all did all of that before, and things could have failed in any place before, including biases in the design, including interferences or some kind of problems in data collection, including inappropriate models and inappropriate assumptions. Sometimes when people tell me this adjustment for multiple testing doesn't work for me, it's really a sign of something else. That something along the way, either you just didn't have enough replicates, or worse, something went wrong during the experiment. Many times when you don't find what you look for, it has very little to do with adjustment for multiple testing. Sometimes it means that you just don't have enough replicates, because when we test more proteins and more hypotheses, we end up having to be more conservative, and this requires larger sample size. So there may be a question of sample size as well. But usually there are signs of something else. Okay. All right. Hold on. Yeah, go ahead.
Here, what you're saying is that LIMMA and MS stats are not particularly different, both in the small sample size or in the large sample size, but they're better than just doing a t-test.
That's for sure. And the reason for that is that t-test is a very generic procedure which was developed over a century ago, where there was really no particular insight in the properties of the data that we are working with. And LIMMA and MS stats and other methods, what they're really bringing to the community is that they build on the specific properties of these specific methods. And it's always the case in statistics that when you bring in more information, you do better. For example, LIMMA said, well, but I don't have one protein, I have many proteins. We can leverage that. Now, MS stats says, well, these are not missing values, they're censored values. We can actually work with that. And then we have multiple protein features which are expected to behave the same way. If they don't, maybe there's something wrong. We work with that as well. So this is why. Now, if the data are really nice and clean, probably t-test would work very well. Usually these methods help, especially when you have interferences or problems that kind of deviate from the expected behavior.
Yeah, just now we learned that when the sample size is small, the interference unlike LIMMA and the MS stat performs quite well when compared with t-test. And it's just a follow-up question: when should we use LIMMA? When should we use MS stat?
So they are not kind of one versus the other, right? Let's see. You can use MS stats with LIMMA. It's not like one or the other, because MS stats focuses on summarization primarily. We look at multiple features in the spectra, working with missing values, working with the outliers. This is one. And the other thing that MS stats is doing is trying to reflect the experimental design appropriately. Meaning, do you have multiple conditions? Do you have a time course? Do you have paired design? Do you have some factorial experiments? We work on that. If you kind of want to think about this, we essentially focus on the signal. So MS stats primarily focuses on making sure that our estimates of protein abundance between the groups and estimates of differences between the groups and individual estimates of variation associated with the signal are appropriate given experimental design and given the interferences and missing values. And we do it separately for each protein. From the design that we have and from the data that we have, we essentially give this single value. If you think of this as a pooled value, we give a better value which reflects the experimental design and the structure of the data, more so than the t-test. But we still have one variance per protein. So nobody's saying that now we cannot use this histogram to the variance of proteins that MS stats provides. And it is actually quite possible. And in fact, I will talk about TMT next, and that's exactly what we do in TMT. In MS stats, we found that we prefer advocate for more than 3 replicates, if possible. And so we found that if we do the summarization right and we reflect the design right, LIMMA doesn't really bring as much to us. And we did really very extensive evaluations for that. But it does add, extra code, extra place for bugs, right? Extra place for, errors. So we decided that we just wanted to stay with that and focus more on reflecting the experimental design and focusing more on summarizing the features. However, the way the method works, you can totally add LIMMA on top of this. And now that we have all these variances and you feel like, okay, I want to do better, so now we can have estimates of variance, a weighted average between every protein and the overall consensus. So essentially, LIMMA is a very specific part of it. You can add methodologically to MS stats. And TMT does it because we noticed that in TMT, when the number of replicate runs tends to be small for the moment, so we try to help with that. In the base MS stats, we decided not to do this because we would rather advocate that at this point people use more than 3 replicates per group. But maybe, I don't know, we'll see what Mina and Ting say. Maybe we will end up adding this if people really, really insist. So another thing about LIMMA is that it's very nicely implemented in R as well. And you can use LIMMA to analyze some of the experimental designs. But MS Stats has more flexibility for the type of designs that we have in mass spectrometry.
So both LIMMA and MS Stats have normalization techniques within the packet.
That's right.
You don't think they're the same type of normalization?
So they may not necessarily be identical, but they're very, very similar, if not.
So either is fine.
That's right. That's right. Yeah. But I would really say that LIMMA doesn't do anything with respect to outliers and censored values, and that's really, and it is fairly limited in the type of designs that you can use. So, for example, it would be very limited if you have repeated measurements design, and it would assume that the type of biological variation which comes from the same person being looked at multiple times across time is constant across proteins, which to me is a very strong assumption. So the way they did it, it's very nice for the variance, but it limits the type of designs that you can model.
I have a question. I think you already said something about it. So about the TMT, we use the relative abundance. So the LIMMA and the MAP status can use that. Another question is that sometimes we have some unbalanced sample. So for one group we have a lot, for another group we want to add a little. So what do you think will influence the result?
Right. So TMT, hold on to that thought. We are getting there. For the unbalanced, it's actually no problem at all, right? So you see that here, the consensus variance, it's a weighted combination of variance in the first group and variance in the second group. So essentially, if you have one group which has a lot more replicates, it will have a much larger weight in the variance. And that's the same for MS stats as well. No requirement of balanced design.
Okay. Another question is if we have paired sample, so we should always consider paired t-test, or we would like to do the two-group t-test?
Yeah, this is also something we talked quite a bit about last week. Paired design is a very good design, just like as a time course, because the person is its own control, right? So let's say you have a tumor biopsy and a healthy tissue biopsy. You really want to look at the changes within the same person as opposed to averages across all people. But this requires, on one hand, to have this design. And if you came here last week, so in the paired design, a person is a block. So the person will look at both sources of variation. And it requires to have the design this way. And second, it requires to analyze the design, analyze the data according to the design. Because if your design has this paired structure where each person has 2 measurements, but you don't analyze it accordingly, you pretty much lose the advantage of this design. So yes, the answer is that if you have time course or paired experiments, you're much better off analyzing this as such. And this is, again, what MS Stats would do. When you say, in fact, we call it time course, but paired design fits the same model as the time course. And MS Stats would recognize this by seeing that you have the same identifier of biological replicate in multiple groups. You have healthy and disease, and you see it's the same person in the two, and MS Stats will fit the right model for that. And so in LIMMA, you'll have to set it up a little bit more, in a more complicated way. You have to kind of specify a little bit how it works.
I have seen the publication from the Bouliens.
From where?
From the lab of Bouliens in Odense in Denmark. They have also published some code where you could use LIMMA and rank product. And afterwards, the p-values were adjusted, and the volcano, not all the dots moved away from the 0 axis. And I tried it on my own data. It was a heavy regulated data set that moved even more away from the 0 axis. Do you have any idea?
Well, this is why. So let me just kind of review this once again. So what happens, right? Let's say you have a small sample size, and your estimates of proteins of protein variance can be kind of larger or smaller everywhere. Now, just to remind you what p-value depends on, right? It depends on the difference between the groups, right? But it also depends on the denominator. If the denominator is large, is small, right, then the signal to noise is large. Your observed values will be very far in the tail of the distribution, and the p-value will be small. So essentially, you can make the p-value as small as you would like if you make the denominator as small as you would like. Now, one way to make the denominator small is to increase the sample size. But another way to make the denominator small is to make the variances small. Now, how do you make variances small? In this moderated situation, your variance is a weighted combination of your protein variance and the average variance. So if your average variance somehow from this curve fitting became very small, all your significance will go up. But the price for this is the assumption that all the proteins have essentially the same variability. So your p-values go small, your significance goes up, but underlying that is the assumption that every single protein has the same variance, because this apparently has a lot of weighting. Is it a good assumption? Well, it depends on the biological system.
With the cell culture experiment, I would rather say we have similar variance than
It depends how the protein is regulated, right? So sometimes very small changes in abundance can have really strong biological effects. Sometimes very large changes don't do anything, right? It really depends on what protein you quantify. It also depends on if it's a targeted experiment or a profiling experiment. Maybe you select your targets very carefully so that they all have the same variation. This could work, right? Or maybe you have a DDA experiment with every protein, and then it can be anything, right? So this is kind of what I mean. We're trying to minimize the assumptions that we make. And that's the reason why we didn't really include this in MS Stats, because it's an extra layer of assuming things. And I'd like to say that if you assume enough, at some point your results reflect your assumptions a lot more than they reflect your data. And we kind of try to make the data speak as much as possible. So that was our philosophy. But again, methodologically, there is no problem at all to add this on top of what MS Stats is doing.
In TMT data using MS Stats, for TMT data using MS Stats, how you do normalization using the peptide?
Okay, so since you're all asking, I haven't even introduced TMT yet. Hold on. So, okay, in this case, okay, first of all, any other questions on LIMMA? We do have time, so let me do one more thing before we get to TMT. And then we will spend, I'm guessing, the rest of time for TMT. So just very small comment. I want to also follow up on the similar type of thing that people use for mass spectrometry. Also motivated from gene expression and microarrays. So the next iteration of transcriptomics, of course, was RNA-seq, right? And we moved on to different type of experiments. And one side effect of these different type of experiments was that the data which are acquired in RNA-seq, they're not on the continuous scale, they are on a count scale, because we count transcripts. And it turned out that when we work with count data, the properties of the data are quite different than when we work with intensities. In particular, the assumptions of normal distributions are not verified. And what happened in the transcriptomic community is that researchers tried to build on things such as LIMMA and other things to now work with count data. And what I see now is that two things. One is we do have count data as well in mass spectrometry when we work with counting MSMS spectra. And in this case, sometimes people don't kind of work with the data as they should with counts, but they use something like t-test or something else. I would like to comment on the fact that if our data are counts, they should be analyzed as counts. On the other hand, what I also noticed is people say, well, there used to be LIMMA for microarrays, now there's DDA-seq. Why wouldn't I use DDA-seq for LCMS, for chromatography-based quantification? And that is totally wrong, because even though we have many genes or proteins, if we use with peak intensity data, we have continuous data and not count data. So essentially, sometimes there is a misunderstanding of what are the properties of the data that make the method appropriate for our particular experiment. So I just wanted to comment on this real quick, and then we get to TMT. So this is an example of different workflows. So far, we spent a lot of time talking about something like that, where we have chromatographic peaks per peptide or per peptide fragment per sample, right? So this is another way, which I hope you're all familiar with, when we have a MS1 peak in the spectra. These peaks are selected for fragmentation. and more abundant peaks are selected more frequently for fragmentation and less abundant peaks. Therefore, the counts of identified MSMS spectra can be viewed as a proxy of the abundance of the analyte. And especially in early days of mass spectrometry, people use this for quantification, in large part because you don't need much computational infrastructure for that. You don't need to find peaks, quantify peaks, and so on. You can only work with database search tools and use this for quantification. And occasionally it is still used sometimes. In particular, for the ABRF study that I showed on Monday, quite a few people still went with spectral counting for quantification. Well, just to, again, repeat that count data are fairly different from continuous data, but it also depends on the range of the count. So here's one example. Poisson distribution would be quite common distribution for count data. Poisson distribution has only one parameter, and when the parameter is large, if you look at the histogram of these values, I just literally simulated Poisson data with parameter 12. It's fairly symmetric and looks like a bell-shaped curve. It's actually quite appropriate to say that this is approximately normal. However, when we have a very small parameter, which would correspond to a low protein abundance, then clearly the counts are skewed. Their discrete nature is a lot more apparent, and the fact that they cannot become negative, it's also more apparent. The problem for us is that we cannot really afford choosing, okay, this protein is in this situation, this protein is this situation. I would say that in every experiment, either RNA-seq or mass spectrometry, we would always have proteins with low counts. And therefore, we have to optimize for that and work with methods which would accommodate appropriately proteins with low counts, even though some proteins may be in a situation like that. So another thing that people would do is say, okay, why wouldn't we take a log transform? We just said earlier, right, that if we have a skewed distribution and we take a log transform, it looks more normal. Well, it's really a problem with this data because log of 0 is not defined. What do we do for zeros? We replace with a small value. The exact value will affect our assumptions. This is not good. Plus, these are discrete values. There is nothing between 1 and 2. When we take a log transformation, we lose this type of properties of the data. Okay, a better approach is to actually work with the distributions which reflect the properties of the data as they are, right? So counts, skewed, positive values, right, discrete, and so on. Poisson distribution is limited because it has only one parameter. A better way of working with count data is to use a negative binomial distribution, which essentially is a more general approach, general distribution than a Poisson distribution because it has two parameters. And here I just have some examples like from Wikipedia. I just lifted different shapes of negative binomial distributions. Different shapes correspond to different values of the parameters. So this is Poisson, and you see that the value of parameter governs that. But negative binomial has more flexibility because it has two parameters. And for some combination of parameters, it looks kind of like normal. But for others, it's a lot more discrete and a lot more skewed. And this is what the genomic community decided to use when working with counts of transcripts. And this is what can also be used, in my opinion, for counts of MSMS spectra. And I will just very briefly tell you the kind of the insight between that. Negative binomial distribution can be represented kind of in the same way as the normal distribution, so in terms of the mean and in terms of the variance of the distribution. And this is how it is done. In this case, the normalization is actually not done before for the same reason I just mentioned, right? If we start dividing this or subtracting constants, we disrupt the discrete and positive nature of the data, so they build normalization inside of the mean. So this is what they call a library size, which is essentially the number of all counts in the entire run. And this is the mean count in the population for the corresponding gene. And this is a parameter which reflects the variability. So it's called the dispersion parameter. And it's also specific to the gene. And this is actually developed by roughly the same community that worked on LIMMA. And they try to do a similar thing. So they will try to express the mean as function of complex design. So again, if you have time course, paired designs, and so on, they will express this appropriately. And they would also combine the information on the variability across all the proteins to have some combination of the consensus variance versus gene-specific variance. In the interest of time, I will kind of move on. Let me just show you some results. So it happened quite a bit, actually, in the ABRF study that people used counts of MSMS spectra, and they used some version of t-test on that, which is completely wrong. And I hope by now we can see that. And this is, again, this IPRG data set comparing condition 1 versus 2. And we can see that the t-test really doesn't do very well, primarily because the assumptions of normality and continuous values do not hold at all for this data. And DSIC could do a much nicer job working with count data. And so this is kind of to follow up on the question before. This is using unadjusted p-values. And we see now that with the adjusted p-values, t-test didn't find anything. DSIC had false positives, so we have only 6 spiked proteins that found 13. And the reason for this is that this type of work with count data requires quite a bit of approximations to reflect complex designs, and also has this moderated aspect that the variance is assumed similar between proteins, and so it has some false positives. However, it is definitely better than using t-test. So this is really the idea. The kind of summary message here is, if we have count data, they should be analyzed as count data, and we have to be careful with the assumptions. If we have continuous data, we should not analyze them as if they're count data, because there's really no point for that. Okay, any questions on that? Let me just take a moment. All right, TMT, finally. Okay, so I may have mentioned previously, this is the first time we present our work on TMT. This is a beta version of MS Stats for TMT. Ting, who is here, can you wave, is the author of that, and she will be leading the hands-on part. What I would like to do is to illustrate a little bit the philosophy behind TMT and why it is different from the rest of MS Stats, and why we could not incorporate this in MS Stats directly. And then after the break, Ting will actually give some examples with hands-on part. Okay, so TMT is a fairly different type of experiment. So it starts with the experimental design. Let's just talk about that. A step back from last week, I will just bring in the slide from last week, why labeling is useful. Remember, we were talking about the fact that when we have sources of variation which are known and large, but we cannot control, it is useful to adjust the experimental design to account for the sources of variation. And blocking was one way to do this, and as we just had a question, so for example, if a person-to-person variation is large, it is actually quite a good idea to have both healthy and disease, or time 0 before treatment, time 1 after treatment, for the same person, because we can make comparisons within the person. And this allows us to eliminate the person-to-person variation. This is blocking on biological variation. At the same time, oftentimes we have a lot of technical or technological variation, for example, due to mass spectrometry runs. And from the quantitative perspective, it may be meaningful to mix together within the same run samples from different sources and acquire them together. Because then, if we have large run-to-run variation, it affects both samples from both sources the same way. And we can make comparisons within the runs, and it will allow us to eliminate run-to-run variability, provided that all the extra steps of sample manipulation and so on don't introduce additional interferences, which is possible. We talked a little bit about that for the targeted experiments with labeled reference peptides. Let me just show you this very briefly. So this was an example of a SRM experiment where, let's say we have one peptide per protein of interest, and we added to the sample peptides which heavy labeled reference peptides in each run. They're added in as constant concentration as possible. So in other words, these heavy labeled peptides, they have no biology in them because they're all spiked in a constant concentration. And so now within each run, we actually have 2 peaks, one endogenous peak, which has the biological variation, and one reference peak, which is only has technical variation. And so now the data that we'll have would look like this. We will have a blue dots, these are endogenous peaks from the healthy group. Red dots, these are endogenous peaks from the disease group. And if we didn't have any labels, we would be comparing the mean of the log intensities of the healthy to the mean of the log intensity of the disease. And if we have a lot of variation, then we may not be able to say that this difference is systematically larger than the variation expected by random chance. However, we actually have this labeled reference peptides spiked in each run, and these black dots correspond to these reference peptides that we have. And remember, these black dots have no biological variation, only technological variation. So in this cartoon, I have quite a bit of run-to-run variation, because the peaks are expected to have the same abundance, but actually they are quite variable. However, these reference peptides, they are spiked in the same run where we have the signal from the endogenous peak. And what we can do, we can compare the deviations of each endogenous peak from the corresponding reference peak within each run. And we see that these deviations are systematically larger for disease than for healthy. And so now we can compare, instead of comparing the means of peaks, we can compare the deviations. And we see that on average, these deviations are larger. Therefore, this protein is upregulated in the disease group. And the way we actually do it, we specify the linear model, which would account for the sources of variation. And it's a generalized way of comparing these deviations to these deviations. This was one way to use blocking. Another way to use blocking is SILAC, where instead of adding a reference peptide in constant concentration, we mix two endogenous samples. And we have now two peaks because one of the two mixtures has heavy label attached to this. And we now make comparisons between two endogenous samples within the same run. And yet another way to do this, and this is what TMT would do, is to multiplex even more. And we can have, so I have here, it's an old figure with the eye track, but TMT looks exactly like that. TMT stands for tandem mass tag, where we can attach labels to the peptides from multiple sources. And unlike SILAC, where we have just two channels, we can have 6, 8, 12, even more potentially channels. And the important difference, so on one hand, we can mix samples from multiple sources within the same run. The important difference is that at the level of MS1 peaks, all of these samples generate the same precursor. The difference in quantification occurs at the level of MSMS spectra, where now each of those different sources, they have different reporter ions. And the height of the peak from the reporter ions inside MSMS spectra, this is what is used for quantification. And this has really fundamental differences. This is why we could not use it in MS stats before. And the differences are just from the data processing perspective. We don't have chromatographic peaks anymore. We have MSMS spectra. So we have to quantify essentially those reporter ions. But most importantly, in order for us to have this quantification, this precursor peak has to be selected for fragmentation, and the MSMS spectrum has to be identified. And this introduces a lot more stochastic variation in the data than any type of experiments that we had before. Because now, the fact that the precursor is selected for fragmentation, this is something which is determined by the instrument while acquiring the data. So we cannot necessarily go back and computationally make sure that every run has the same precursor ion fragmented. This is something which happens experimentally inside mass spectrometry. And then we have an extra challenge that we need to identify the MSMS spectrum and establish the fact that it belongs to this protein. And this is something which requires the presence of MSMS spectra in each run. So unlike in MS1 spectrum, when we can propagate the intensities from retention times and so on, here we really need to work with MSMS spectrum. So the upshot for us is that we cannot rely on the presence of exactly the same peptide ion in every single mass spectrometry run. We may not have data for the same protein in every run, and if we have data for this protein, it can actually come from different peptide ions in different runs. And we have very little control on that, and we have to work with the fact that we'll have different peptide ions in different runs, and we will have missing ions in some runs. On the other hand, the positive aspect is that we mix more samples in one run, and if there's an artifact, for example, intensity went up completely or went down completely for some technical reasons, it will affect the same way all the reporter ions. And comparing within the SMS spectrum would allow us essentially to eliminate the variation between these conditions, which is due to inconsistency of ionization and so on. Now, so this is really difficulty on the experiment, on the data acquisition side. And there's another difficulty which relates now to the experimental design. The fact that in label-free LCMS we have one run per sample makes things very easy in a way, because we have a sample, we acquire the data from it, right? And if we have more replicates, we have more runs. If we have more conditions, we have more runs. There's really not much to do, well, besides maybe thinking about randomization strategies and things like that. If we have now multiple channels, we have a lot more choices to make in terms of how we allocate biological and technical replicates within the same run versus between runs. And what I would like to introduce now is a controlled mixture which we will work with for the hands-on part. And as I mentioned before, we love controlled mixtures because we know the truth, right? We know what are the abundances of proteins that we have in the mixture. We will pretend we don't know it, and we will try to uncover these changes by analyzing the data. And so this is one particular controlled mixture that we have. So we have, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 channels in this particular data set. So this is 10 TMT channels. The way the experiment was designed, this particular version of experiment was designed, is that we have different concentrations of a protein in this mixture. So just as before, we have a complex background, and we have multiple proteins spiked in this concentration. So for a particular protein, we have 4 different concentrations here on some arbitrary scale. And the characters inside, these are the channels to which we have this particular concentration assigned. So we see that this concentration is present in 2 channels, this concentration in 2 channels, and so on. The last 2 channels, or rather the first and the last, are used for normalization. So these are the 2 channels where we will have the same concentration across all the runs. So now already we see that we can choose whether we have different channels having different conditions or have multiple channels within a run have the same condition. So here the concentration is essentially our condition. In this particular case, we decided to look at the variability within a SMS spectrum and have 2 channels having the same condition. From this point on, we can have multiple mass spectrometry runs. In this particular example, we literally acquired 3 mass spectrometry runs of the same mixture. Therefore, the only variability between the run is literally due to mass spectrometry. There is no biological or kind of mixing variability due to this. And then we did something else. We did more. We said, well, maybe the way we labeled the samples introduced additional variation, or just the way we created mixtures. So we repeated this experiment several times. Now, it is exactly the same structure as before, but with the differences that in each mixture, we have different channels allocated to different labels, and each mixture was made from scratch. So within the mixture, the only thing that varies is different mass spectrometry runs. Between the mixtures, the actual manipulation of biological material varied. A problem with controlled mixtures is that we don't have biological variation. And so the fact that we label things differently every time for every mixture try to mimic a little bit more variation introduced by data manipulation. And then the question would be, okay, we have multiple proteins, we have background proteins, we have proteins spiked in these concentrations. Can we now look for changes between these conditions across runs and across mixtures and see which proteins are changing in abundance and which proteins are not changing in abundance? So the first reason why TMT is more difficult is because we have a lot more options in terms of how different sources of variation are allocated to within MSMS spectra, to different runs, and different mixtures. And this requires quite a bit more work on the statistical analysis side. But this is the first thing. And then the second thing, as I mentioned, the fact that MSMS spectra are not present in every run. So not only we have differences in labels in different mixtures, but also the actual ions that we quantify in each of the runs may not necessarily be the same. Yes?
So in this example, you have 4 conditions.
That's right.
And what if you end up having like 12 conditions and you only have
Hold that thought. I'll talk about this a little bit later. Exactly. So these are exactly, and so when we try to design software which will be useful for a variety of experiments, we have to consider all of that, right? So what if I have fewer conditions than channels, then I can have replicates within MSMS spectra. What if the number of conditions is exactly the same as channels? What if the number of conditions is larger than the number of channels, right? And all of that changes the stochastic structure of variation in the data, and it ends up being an effort completely different from what we talked about in MS1 or chromatography-based quantification. So as I said, this is our first kind of venture in this, so we'll show you how we think about that. We'll show you how far we got. Hopefully it will be useful for that. Well, as in the case of MS stats, we do not do peak identification, peak picking, peak quantification, all of that. We rely on the work of people who do this better than we do. In this particular case, we have converters from MaxQuant and from Proteome Discover. If you have data using TMT where the peaks are identified and quantified with these tools, we can export the data into MS stats. We're also working actively with OpenMS, so I think OpenMS will be coming very soon as well. And in the future, we will probably work with other tools just the same. For the moment, that's what we have. Let me now contrast the structure of the data for MS1 with the structure of the data with TMT. This is what we had before. We had multiple conditions. Within each condition, we had biological replicates. For each biological replicates, we could have technical replicates, and then each technical replicate was a run. And this was the biology, right? And we also had a technological aspect where each protein had multiple features. So there's several important things, right? So we have the same feature across all runs because in, for example, SRM experiments, we can target exactly the same ion in all the runs. In DIA, we can extract the fragments across runs for the same M over Z. And in LCMS, we can identify MS1 peaks in some runs, and we can propagate the intensities by matching mass, overcharge, and retention times. So usually, in chromatography-based quantification, we can ensure with some more or less missing values and so on, but we can have the same features between the runs. What we also have is that the restriction on randomization occurs within the run. The conditions, the way we acquire data on the conditions can be randomized. The subjects can be randomized, and the runs can be randomized. The only thing that is not randomized is how we acquire features. So all the features are acquired simultaneously within the same run. So now let us contrast that with what happens with TMT. So with TMT, remember, within the run, we have to acquire things simultaneously. So everything starts in a run. Within the run, we have a restriction on randomization. We can randomize the subjects, which we can allocate to the runs. We can randomize the order in which we acquire the runs, but then all the features will be acquired simultaneously within the runs. So now the run is a restriction on randomization, and we have potentially multiple conditions and maybe multiple biological replicates, like we just said. If we have few conditions, more channels, we can even have multiple biological subjects within each run. So this is a big difference in the structure of the data. And the other difference is that the features which we acquire in run 1 are not necessarily the same as the features we acquire in run 2 for the same protein, right? We can have a different charge, we can have a different peptide for reasons of fragmentation, but also for reasons of identification. Maybe we could not successfully identify some features in this run, which we could identify in this run. So essentially, the stochastic structure of the data is different. And the way we approach this, that we essentially felt that there is no sense saying that feature 1 here is the same as feature 1 here, because it's not. It's not necessarily the same ion. So what we will do, we will work on the summarization of features separately for each run first, so that we have one number per protein per run in each condition, and then we will work on the normalization and statistical analysis. So this is what we currently do. So essentially, from this run, we will use the same approach, censoring and Tukey median polish, to account for outliers and missing values, to have one number per protein in this run. So it will be here. And then we will summarize this data structure. We'll have one number per protein in the second run. We will have run number 2 here. And so from this point on, at least, we eliminated some of the differences between ionization and between the runs. And so the next step, will be the normalization of statistical analysis. Before I get there, here's an example that I think we'll be showing to you during the hands-on part. Remember, we looked a lot at the profile plots in MS Stats, right? So now these profile plots are broken into panels, where each panel is a single run. So this will be the profile plots for one protein. Each panel is one run. And within each run, we have multiple channels, and each color is a different peptide ion. So for this particular protein, you see that, for example, here we only had 3 peptide ions. Here we had a lot more peptide ions identified. And you see that the colors are not consistent. For example, here red indicates one peptide, which is clearly missing in these runs. So usually, the peptides which are consistently profiled across all runs are very rare. So we really have to work with peptides which are very different across different runs. Then if we use this summarization so that we have one number per protein per run, using censoring and using robust summarization, we will obtain summaries which look like that. The gray lines here, these are different ions, and the color line is the resulting summary. And now we have this. We have a much simpler data structure where at least we summarized all multiple MSMS spectra into a single spectrum. And at this point, we can work with statistical methods which would account for various allocation of subjects to conditions and runs. But before we do that, we need to normalize, because still run 1 and run 2 may be based on summaries from different spectra. And this is the example of normalization. Ting did quite a bit of work trying to understand if, for example, some versions of shifts, median shifts, or quantile normalization or so on will be helpful. In our experience with this type of data, using reference channels were really helpful for accounting for differences in type of ions that we have in different runs. So the idea here would be that we would equalize the intensities of reference channels between the runs, and will apply the same shifts to the endogenous intensities in the corresponding run. So using reference channels for normalization after summarization. And again, Ting tried to do this before, and we found that doing this after summarization gave more accurate results. And then we would be working with this type of data structure after normalization, where we have multiple runs, which are essentially blocks, creating a restriction on randomization, multiple conditions, which we want to compare, and then we will have to account for the fact that sometimes we have biological replicates within conditions. Sometimes one condition has one biological replicate. Sometimes runs are technical replicates. Sometimes runs are biological replicates if we have larger number of replicates. And this is something that MS Stats can work with, because MS Stats builds this very flexible mixed effects models which account for really complex experimental designs. And here's one example in a particular case. So let's say we have multiple conditions and multiple runs. And if we have in the condition, let's say we have technical replicates for the same subject, then we can essentially decompose the intensities, the variation in the summarized intensities in each run into the contribution of conditions that we're interested in, the contribution of blocks, which are runs, the contribution of individuals if we have technical replicates, and then random variation. And in some special cases, so we can derive even the variability associated with the estimates between different groups. And we can now use this to plan experimental designs to see, for example, what happens if we have large between run variation versus small between run variation, and so on. Now, going back to your question, we can also say, okay, if we get that far, we have these models, we have variance components. Now we can actually estimate these variance components or project these variance components for different allocations, including what if we have more conditions than labels, right, and so on. And this is where, in fact, we can go back also to the genomic literature, because it also, some 15 years ago, there was similar work with microarrays. I don't know if you remember, there was very early version of microarrays were CDNA microarrays, spotted microarrays, which had two colors, right? And people were mixing two colors, like spotting arrays on the same plate. But from the statistical perspective, it was very similar, because now we mix together samples from two sources. We have a limited number of channels. How do we allocate samples to the channels? And at that time, there was a lot of work on comparing different designs with this type of limited resources. And it turns out that we can leverage a lot of statistical methodology from this. And let me just give you some examples. So this would be, and this, by the way, is from the review I wrote with Anne Oberg quite a while ago, which talks about this specifically. So at that time, it was in the context of iTrack, but now we can reuse the same methods for TMT. So if run is a block, meaning that run gives us a restriction on randomization, a very good design would be, let's say we have 5 disease groups and 5 channels, just have one biological replicate from each group in one run, another set of biological replicates, one from each group in another run, and so on. However, if we have 4 labels in 5 groups, then we're stuck, because we cannot have one replicate from each group in 4 labels. There are many ways to allocate 5 groups to 4 labels, right, and replicate runs. And we can use essentially the same approach here with the linear models for each design and calculate the extent of variation when comparing the groups from each design. And it turns out that some designs minimize the variation more so than other designs. And in particular, this particular design, which is called balanced incomplete block design, we can show that it is more effective in terms of minimizing the uncertainty in our comparisons than some other designs. So this design will have a very systematic rotation, so that in each run, so here block is a run, always a different disease group is missing. So you see that in here group 5 is missing, here group 4, here group 3, here group 2, and here group 1 is missing. And now it will take 5 runs to systematically rotate this type of allocation. But if we do that, as opposed to just randomly or somehow in some other way allocate the resources, we can show that the variability in our comparison will be smaller. So our experiment will be more effective. And in old days, when we had only 2 channels, and this would be an example of SciLAC, for example, we can extend this the same way, where we always have different pairs present and different combinations are missing. This gives us constraints. For example, here, to have a systematic type of structure requires at least 5 runs. And in fact, if we want to have more replicates, we have to have multiples of 5. And here, if we have 5 disease groups and 2 labels, we will have to have 10 runs in this type of systematic structure. And the more replicates will have to be multiples of 10s. But in terms of the uncertainty that we can characterize, we will be more effective than if we use, for example, a single reference channel and just compare everything to the reference channel. And in our review, we showed like different variance components and how it works. And this is the type of a consideration that we can have. So, of course, there are different costs associated with this type of allocations. And there's a cost and feasibility and so on. But there are also statistical considerations which experiments will be more effective. And the good news is that this type of models which we develop, they actually work with a variety of allocations. So you can essentially feed the same type of model for different allocations of subjects to groups, but the numbers will be different depending on the allocation. So this is a very kind of initial summary of TMT, and I'm sure you will have questions. Let me maybe stop somewhere here, and let's see if you have questions about that. Yes?
So you showed the incomplete balance and randomized model design. So you're taking 5 disease groups, and now you're running 10 runs, or 5 runs. Well, 5 runs for the 4 channels.
So this is called a minimum. So okay, so here are 5 runs. This is called a replicate set. 5 runs will ensure that each disease group has the same number of replicates.
So my question is there any benefit in terms of the statistics and minimizing variance than just taking those same 5 and doing a single shotgun DDA?
Oh, okay, so that is not a simple question, right? Because what we do here in terms of variance components, we assume that, let's say you allocate samples to channels like this versus in some other way, your manipulation of the samples will give you the same type of variability, right? What you are asking is essentially label-based versus label-free. So what I can do, I can tell you variance components of this design versus 1 sample per run. But to calculate numerically what it would look like, you would need pilot study of both to understand how much extra variability is contributed by extra sample manipulation from labeling. Mathematics can take your pilot data and say, if these are your variance components, let's say from TMT, I can tell you how much sensitivity I will have for different sample size. I can also tell you how much sensitivity I will have for different allocation of the same number of samples to different channels in different ways. However, to compare this with label-free, I would need very different variance components from label-free, because it's not just mathematics, it's also how much interferences you introduce by essentially labeling process, right? But also, for example, interference at the precursor level can change variation, right, in TMT as compared to MS1. So these are fundamentally different processes experimentally, so they would have different variance components, even for the same biological sample. So I would need to have pilot data from both workflows to really make an effective comparison. Within here, I can use the same variance components to compare different allocations of samples to labels, assuming that for all the allocations, the interferences will be of the same type on average.
Yeah, because oftentimes when I think about using multiplexing strategies like this, it's to minimize the number of runs.
Of course. But you can introduce variation at the MS1 interference level, at the labeling level, right? And of course, there are other issues, as always with pooling, right? If you have 1 sample compromised, the entire pool is compromised. If you lose 1 run, you lose a lot more replicates. These are very kind of multifaceted considerations, right? But the way statisticians can help is to make projections, right, in terms of how much variability you would have. But usually, because these are very different data acquisition workflows, which have very different technological variation, you would want to have pilot studies from both and compare using variance components from each separately. Oh, so this is chemical labeling, and you would need to talk to someone who does these experiments. Maybe somebody can comment. So these are essentially chemical labeling of the samples as opposed to metabolic labeling in SILAC. And these labels introduce and shift not at the MS1 ions, but in the fragment ions that you could see. So I'll let maybe experimentalists comment on that more.
I have some slides.
Yeah. Maybe you could do during the break, you could do a little bit of an intro in TFT. Maybe you can have a poll and see who would be interested in that. Can I see a show of hands who would be interested in learning more about the experimental aspects? Or everybody knows? Looks like, well, maybe in the morning or during the break if someone is interested. Let's talk about more statistics, yes?
Just want to clarify this experimental design.
Yes.
You have made reference channel, right?
That's right.
Can be used eventually for overall normalization.
That's right.
Initially, you are doing each run-to-run normalization, and then you are using the reference channel for all normalization, or?
No, normalization is tricky, right? Because you don't want to over-normalize your data. So you really want to do just as little as necessary, but not more. And we decided, based just on empirical evaluations, take this data not normalized. But here you don't need normalization within the run, right? Because every row here comes from the same MSMS spectrum. So for example, if an SMS spectrum was selected for fragmentation at the beginning of the chromatographic peak, it will have low intensities altogether, right? If it was selected on top of the chromatographic peak, it will have high intensity. So first row will have low intensity, second row will have high intensity. It's okay, because they were all within the same run. And now what will happen is, well, you can think of it just as some kind of average for this channel across all features, average across, and so on. And these averages are comparable because they're based on the same features, right? So now they're not comparable between runs, but they're comparable within runs. And that's all we need for the moment. So essentially, we treat each run as a separate experiment and summarize. And because it's a separate experiment, which is all comparable because it's all acquired within the same run, so you don't need any extra normalization. We go straight for summarization here. But then we have multiple runs, and this is where we need normalization. And so the way we set it up was that we first have one summary per run across multiple ions, or multiple SMS spectra, which were produced within this run. And then we normalize these summaries. And I think this is a better way to do, in part because the summaries are more reliable than individual ions, right? And so those will be normalized between the runs. But now between the runs is difficult because maybe some of this is biology. Maybe run 1 has one set of biological replicates and run 2 has another set of biological replicates. On the other hand, maybe they have different ions. Maybe some ions are selected in run 1 but not in run 2. It is very difficult to make assumptions that on average the signal should be the same or something because they're very different things. And therefore, the reference channels are really helpful here. And they, by the way, are helpful in many ways because also in terms of making sure that MS1 peaks are abundant enough. If you have a reference channel with enough biological material, then the precursor will be abundant enough and selected for fragmentation. Because what can happen is if the protein is missing in all the conditions, you may not see it at all, right? But with the reference channel, it ensures that the precursor peak has enough biological material in there. And then for normalization, then we know that the reference channels are constant. And it is much more appropriate to normalize those. And that will account for difference in ionization between the runs, and things like that, but will not mess up with the biology that different runs have different biological abundance.
So here, what normalization scheme you apply from this?
Sorry?
From this reference channel, how you do normalization?
Oh, so here's an example, right? So here's reference channel has intensity 10, 11, and 12.5. So now to 10 we add 1 to make it 11. To 12.5 we subtract 1.5 to make it 11. So now we add +1 to every single. To 3 we add 1. To 5 we add 1. To 6 we add 1, right? And here we subtract 1.5. So now from 5.5 we subtract, and we subtract as well, right? So you choose the median. So here 11 is the median, right, of these three numbers. So you add a constant to the first run so that it becomes the same as the median. You subtract the constant from this run so that it becomes the median. And then you add or subtract the same constants to all the endogenous values to make them equalized. So you learn these constants on the reference and you apply the same constants to all the endogenous. And again, this is on the log scale, right? So we do averages, add and subtract on the log scale, right?
There are also some bioinformatic approaches to the ratio conversion in isobaric data. We also have something included of that.
What do you refer to? Can you give more details?
So the ratio distortion. So the ratios are usually in isobaric tables.
Ratio of what to what? Ratio of what to what?
So if you compare two channels.
Okay, so, and that's kind of the similar theme as in MS Stats in general, where people compare conditions first or channels and do some things to them. Statistically, the correct approach is actually the opposite. First, prepare your data, and then compare conditions. So we get to the ratios or to log-fold changes at the very, very last step of data analysis. So if you do the summarization right, and you do the normalization right, then you use the summarized, the normalized data to have these generalized ratios, right? Compare the fold changes between conditions. And the reason for this is really kind of philosophical, right? If I go at the very, very beginning of that, what we had in the slides, this is how it works, right? We collect the data, we look at the signals which has the right statistical properties. And from the signal, we infer the differences in the populations. So everything we do in terms of summarization and normalization is making sure that the statistical properties of the summaries are as compatible as possible with the inference we try to make. And what you describe is first take the summary and then look for fold changes, and then manipulate the fold changes, right? Philosophically, from the statistical inference perspective, it's the opposite. You transform, normalize, summarize the data so that the assumptions are verified. And then you take the summaries from the transformed data, and these summaries will be optimal in the statistical sense. So taking the ratios first is essentially not optimal in the statistical sense. First you prepare the data, and then you compare conditions, and not the other way around.
Then you assume that your variance would also shrink with the ratio of the distortion.
So I think I have a similar question.
Mm-hmm.
There's a couple of papers saying that isolated ego, if you isolate a peptide, the peptide are whole species that give the water ion will distort the lower abundant ion.
Well, okay, so
So basically, isolation means so you can have two peptides. One is very abundant, the other one is very low. And for example, the low abundant one will give out, let's say, 2 to 1 to 1 to 1. But the housekeeping team will have 1 to 1 to 1 to 1. And the 1 to 1 to 1 will distort the low abundance ratio. So actually, I have another question. So from the protein discoverer, they have a way to set this up. So I don't know how to
I think Ting will be able to show you some, to have some discussion of how to work with Proteome Discover for that. But let me maybe also put something else out there. Clearly, the interferences, right, at the MS1 level are important, right? And this is something that happens at the experimental level, right? It doesn't really happen at the kind of bioinformatics level. So this is part of how the data are, right? We can try to correct for that using some assumptions. And every time we do something with the data, we make assumptions. Either we acknowledge this or not. But every time we take ratios or differences or do something, we make assumptions, right? We just may not necessarily spell it out. I would actually argue that if the experiment themselves compromise the fold change, it may or may not be a problem, because our goal is not necessarily estimate the fold change. At least the way we state the goal here is to detect regulation. If we can say that there is a significant change in abundance, this may be enough for us to move on and follow up on those targets, even if the fold changes themselves are compressed or distorted. And so at that point, it becomes a question of how much the fold change was distorted versus how much the random variation between the rounds or between replicates is affected. Because even if your fold change is biased, essentially, right, is reduced, if the associated variation is small enough that you can claim the change, you will be able to follow up on this target. And that's really all you need oftentimes, right, in this type of experiments. I think the reason why people worry about this a lot because they use fold change cutoffs. And we argue very strongly that fold change without estimate of variation makes no sense whatsoever. If fold change is really important for problems like, for example, you want to compare regulation protein abundance with gene regulation, right? And you want to see how changes in gene expression affect changes in protein abundance, then estimate of fold change is really important. Maybe then it's not the best workflow to use, right? Because the workflow distorts fold change. At least the way I have seen it so far, this type of experiments are often used as screening experiments which will generate targets for follow-up. And oftentimes it's enough just to say it's up or down. And it's more systematic than random variation. And in that sense, I think it's fine, right? But if you really want to do like the really accurate estimation of fold changes, if you're doing, so maybe that's not the best workflow for that. So for the moment, we kind of stay with this framework.
So you're basically saying using this approach, you have precision and necessarily accuracy.
That's right. That's right. And it's a combination. Of course, if the fold change is large, it's easier to detect the change. If variance is small, it's also easier to detect the change, right? But in many cases, and even, by the way, in DIA, right, or in, like, the fold changes are not accurate, right? Because also you hit the limit of the dynamic range, right? So there are many reasons when the fold changes are distorted. But for many applications, it may be enough as long as we find that there is a change in abundance. And I think this kind of trying to get the fold changes right is probably motivated by the fold change cutoffs, which in my view is a really wrong approach statistically altogether. Okay, I think it's time for a break.